learning and so on. But we are going to see, I hope throughout this presentation, how we analyze the microbiome. Because when we say microbiome, as you might be well aware, we are referring to a lot of different types of features that we can exploit. As a very quick summary, I'm going to touch upon three main topics. Last one will be FMT again, but from a computational perspective. The first one is that I would like to try to introduce and give you some hints about how we really deal with the analysis in profiling and use the microbiome when doing associations, in this specific case with machine learning, and then its application to the case of colorectal cancer, so how we can define what is a microbial signature, and that colorectal cancer is a very nice use case in this case, and then how we can plug into different type of features from the microbiome, so from bacteria to microcariots, in the association with diet and cardiometabolic disease. So, first of all, when we say the microbiome, I'm usually always talking about shotgun metagenomics. So we take all the DNA from a sample, we do shotgun sequencing, we do computational analysis, we get our profiles, and then we use them for the downstream analysis after that. And this is very important because it gives us access to many different types of features, from genes to bacteria, archaea, fungi, viruses, eukaryotes, that we can survey and provide. And when I say machine learning, what I have in mind here is that we have microbiome samples and we have information about the samples, for example, people that are healthy or people that are diseased, and we can extract features from both of them. So features can be genes, can be pH, gram staining, can be the bacterial taxa, the functional metabolic pathways. Again, we can also, from the information about the samples, we can get our target values. So in this case, we can say whether it is a CRC or a healthy individual. And then we plug this into what we can say machine learning, which we separate into a training phase at the beginning, so where we use these features for all the samples and the target values, and we train a model that we can use. So the model will be trained to try to predict, to link these two entities. And once we have our trained model, what we can try to do here is, if we have new samples with unknown information about them, is to encode the same features for the samples, apply our model, and make the prediction. And if we apply this to CRC, the idea is that we have fecal samples, we do metagenomic sequencing, and we have relative abundance of the taxa present. We have information about CRC or controls, that's what we want to learn in our model. And if we have a new fecal sample from a new individual, previously unseen, we apply our model, and then we can say whether it is CRC or control with certain confidence. And I say confidence because there are different ways we can measure our machine learning models. So if we have one dataset, we have one cohort with n samples, the basic thing we can think to do is cross-validation, meaning that we will split our cohort into a training set and a testing set, because we want to test our model on samples that were not used during the training phase. And we can think to repeat these test set selection n times. In this case is what we can call as key-fold cross-validation. What you see here will be a 5-fold cross-validation, meaning that we have 5 different testing subsets. In this case, ideally independent. We have the remaining sub-parts of the cohort that we use for training. And so we train our model here, we test our model here, and then we can try to make a summary of all these iterations when we do the evaluation. And I was saying we have different metrics, so we can have, for example, accuracy, we can have, if it is working, precision, we have recall. What we mostly use, though, when we're talking about classification in particular, is AUC, area under the rock cure. And in this case, rock cure is a balance between different metrics. that we can simply translate when we have a value of the UC of 0.5. That means that we are not able to model anything. We have random prediction. If we are close to 1, we have a model that is able to perfectly predict our target values that we want to predict over values. And if we apply this to CRC, a while ago we sequenced a cohort in Milan here with, you see from the legend here, we had CRC and control individuals. Up here, there were no adenoma in this case. We did cross-validation within the cohort to try to see if we can use the microbiome species here, that you see the relative abundance encoded with the code, with the color code here. If we were able to discriminate